Hi, my name is Joseph, and I'm back here to talk about the Keio Collection today. So the Keio Collection to microbiologists is a collection of single gene deletion mutants. What does that mean? Well, for those of you who know, essentially a CAN cassette knocks out a single gene, and so you can get these single gene mutants. So when someone says like delta lam B, for example, that means that the lam B gene has been knocked out, and if you see two colons after it, it is replaced with CAN. So what does that mean? That means that you essentially have a collection of single gene knockouts that you can use for cloning. Why is this important? Well, when we talk about model organisms, E. coli is the go-to model organism. And in fact, in 2006, there wasn't really a model of this scale that people could do functional genomics on. So talking about the construction of the Keio collection, I think is very important for microbiologists to understand, and something that I struggled with early on. So today I'm talking about the construction of E. coli K12, and I'll link this paper in the description below. But really what I want to talk about is the impact that this paper had on the field, as well as how to really use this paper within your own molecular cloning techniques so that you can understand how to get the most out of your molecular cloning. So what some people don't know is that this paper not only was about the construction of the Keio collection, but also that of looking at non-essential genes, in which they talked about later. And to do so, they started out with this genealogy here. They started with E. coli K12, and they looked at two-stepped descendants, MG6055 and BW25113. Something that really stood out to me, like you don't have to remember these numbers, is that E. coli has a lineage that you use in lab, and I think that's really cool because just like we look at our family trees, there is like there is ancestry from E. coli, and I think that's interesting because like even when I was first working with E. coli in the lab, I was not sure if it was pathogenic, but kind of understanding the history of E. coli is something useful to know, and sometimes a reason why your experiments don't work. Like I've had lab mates that have swapped strains because things didn't work. So talking about design of the in-frame single gene deletion mutants. So I'll move this here. That was my practice. But the construction of Keio collection strains was actually quite simple. First off, we started off with the wild type genome of MG1655. So here you would have your gene of interest that you're planning to knock out. So gene of interest, abbreviated GOI, and the molecular technique that we used was lambda red recombinering. And so lambda red recombinering depends on the sequences of homology near the gene of interest, which I'll show in red. Now what we wanted to do is to replace this gene of interest with our kanamycin cassette. Oh, let me change this back to black. We wanted to replace it with our kanamycin cassette. And our kanamycin cassette is simply an antibiotic that we can use to select for successful transformants later on. So in order to do this, what you have to do is make a forward primer and a reverse primer, then PCR this product that has arms of homology at the end of these primers. So these primers are approximately 50 base pairs in size. And after you got this CAN PCR product, what you could do is then add this plasmid that has exo, beta, and gam. So what does lambda red really do? Lambda red is a simple method for doing homologous recombination into the genome of MG6055 and other different types of bacteria. And it's reliant on these three different types of protein, exo, beta, and gam. Now, you don't really have to know detail, but I'll talk a little bit about it. So exo, after we have that PCR product, that double-stranded PCR product, chews back as an exonuclease until you get single-stranded or double-stranded DNA with single-stranded ends. Now, beta protects those ends from degradation, and GAM goes one step further to inhibit the REC-BCD system. Now, the REC-BCD system is a system that is the bacterial DNA repair system. So by inhibiting that, we can allow single-stranded DNA to persist within the bacterial cell. Now, what's important about having this piece of DNA, which remember it would be this canned PCR product, in with a gene of interest, is that it fully replaces the gene of interest. So what you would see in the genome afterwards. Oh, let me make this back to black. Ooh, that's not working out well. There we go. Okay, after we'll get the genome back to black, then we'll have insertion of the CAN cassette. And this CAN here is from PKD13. And PKD13 sequence I have here, you can look at the actual sequence if you want to create a primer, which I actually will do for diagnostic PCR. You can use that, where you download from GenBank, to double check by using a primer from within the CAN cassette, as well as if you remember this little bits of red, since that we've replaced the gene of interest, these little bits of red are now directly adjacent to the CAN. And so you make your primers here, and I'll show your forward primer. Here we go. Show your forward primer, reverse primer. And if you get a band of this size, then you know that you have successfully integrated the CAN into your genome. And you'll see that within their diagram, notice that they have this primer here, K1, and this primer here, U. And these are to double check and verify that they got their kanamycin integrated into the genome. Now you'll notice here there's a little bit more complexity in that there's FRT sites around the CAN. So if I go back to the sketch pad and I show the black, what these FRT sites are in general are directly near the CAN cassette, and you also integrate these into the genome. And what they're important for is after you've inserted the CAN into the genome, you can use this FRT site in a process that takes approximately 2 weeks from personal experience to knock the CAN out. And this is important because the kanamycin cassette might give a slight fitness defect to your strain, or you might want to use kanamycin as a selection marker for a later experiment. And so these FRT sites allow you to basically remove the CAN after you've integrated into the space of the genome where you have your single gene knockout. Something really interesting I think that people don't know about the Keio collection is that in order to make sure that the deletion was in frame, they actually use the start codon of the target gene and the stop codon of the target gene in order to create this tiny FRT 34 peptide long, I hesitate to call it a protein, but like this small chain of peptides. So there's actually inside the cell a small peptide floating around due to the process of creating the Keio collection mutant, which no one's ever really thought about, or I've never actually heard someone talk about, but that's just kind of interesting stuff. And finally, I want to talk about, you can look here to see how they did, how they look at different open reading frames and compare it to other studies that also looked at non-essential genes, and how they found different numbers of non-essential genes than before, and how some of the other papers actually made errors in polarity, which means that they thought that some genes were essential because when they knocked it out, it caused the death phenotype of the cell, because when you knock out something essential for life, you kill the bacteria, when in actuality it was a gene downstream of that gene that was actually essential for life. Something else interesting that I found was that they were able to resolve some really cool problems where the genes were overlapping with really nifty PCR techniques. And they also did this really cool growth assay where they grouped their different single deletion mutants based on their growth in MOPS minimal media versus LB, and found the ones that had growth defects as well as things that improved growth, and for the majority of the mutants did not have a significant growth defect, whether they're in minimal media or rich media. So anyways, that was my breakdown of the Keio collection construction, and I hope you enjoy. I hope that kind of helps you. Just remember, PKD13 is the kanamycin cassette, in case you ever forget. That's where I had problems with. And in case you're ever looking for the primers that they use, you can look in their supplementary material, and they have tons and tons of information that you can parse through for looking at different genes. So anyways, hope you guys enjoyed. I'll see you later. Bye!